Following Carlos is always a tough challenge, but want to focus your interest on the phenomenology of DNA repair defects that exist across the whole of cancer, but in breast cancer in particular. I'm still clean. Maybe that's a criticism these days, but anyway. Yeah. It cost $15 billion to sequence the first human genome. Today, I can order a whole genome sequence from the New York Genome Center for $500. And we get information about mutational burden in cancers, which again, I think this observation is now being hijacked by the immunology field because mutational burden and response to immunotherapy is now one of the hottest topics in the field of cancer research. But what this, I think now, almost iconic figure represents is that mutations are incredibly common in cancer across the entire spectrum, from melanomas that are incredibly high, but even down to glioblastoma and breast cancer, you can see here, have a significant number of mutations. You can drill down, as the Sanger group did, and characterize what they call mutational signatures. Now, I know this, again, this is almost a picture rather than a data to read, but these mutational profiles basically said, we know the entire base sequence of the cancer. What patterns can I recognize? And what it found for breast cancer was that, again, I'll drill down on this in the next slide, but certain signatures, certain patterns of mutations, single nucleotide variant mutations in context could be seen for every type of cancer. And breast cancer had a pattern of its own. And that is shown here. So these are the mutational signature contexts where you have essentially 96 permutations of base pair changes in context, 6 fundamental base pair changes. And if you take into account the neighbor on either side, you can get those 96 permutations. By using that information, you could deduce these different single nucleotide variant mutations. And the ones highlighted in red are those characterized for breast cancer. Signature 1 is basically what's generically called aging. Aging means the number of replicative cycles you've been through is more likely to eventually accumulate an error. Vogelstein's group showed this very clearly, and the commonest error is C to T, because that's just, I guess that's the 10 commandments of DNA repair. That's the commonest alteration that takes place. So signature 1, if you go back to, is prevalent in all cancers. But what I want to highlight is signature 3 and 8, which is this very flat profile. And those are strongly associated with homologous recombination deficiency, which I'm going to use most of the talk to highlight, since that's where most of the interest in repair defects in breast cancer take place. But I initially want to highlight this other phenotype, this signature 2 and signature 13, which have this characteristic C to G, C to T combination. And again, this is a phenotype that's probably not as well known because it introduces this concept that not all DNA repair pathways fix things. Occasionally, DNA repair pathways cause the mutation. And this signature 2 and 13 is an example of what's called APOBEC activation. And many of you in the room may not know what APOBEC is. It's an aberrant cytidine deaminase activity that creates that mutational profile. It's a very, very common cause of mutation in cancer, and it's the second most common DNA repair spectrum in breast cancer. And I'm basically highlighting it now because I think we've not paid enough attention to it. What's its real role? Its real role, like a lot of things, is to protect against virus infection. It actually has already been hijacked in the immunity development because it's what drives somatic hypermutation. Because the AID enzyme, which is a key driver of immune development, is an ApoBec or cytidine deaminase. But this signature is seen very commonly in cancers, and it's caused by when you have single-stranded regions of DNA, which essentially is any gene that's transcribed, the cytidine can be deaminated to a uridine. And when you have a uridine, the uridine glycosylase can remove it and produce an A-basic site. That combination of changes that take place can give you this change in sequence of C-cytidine to uridine, which in DNA terminology produces your C to T mutation, and the A-basic site produces this C to G mutation. So again, this sequence of the repair process acting as a generation of mutation is contributing to this significant DNA repair defect, which is particularly prevalent in breast cancer. And again, this is simply to show these are mutational events that can occur in relation to a schematic replication fork. But what is shown at the bottom here is that deregulation of the ApoBec enzyme creates these C to D, these C to T and A-basic sites in the region of the replication fork that creates a form of replication stress. And the question is, how can we diagnose this? Well, again, you diagnose it by the mutational spectrum. How do you validate it? That actually is still a problem, because it'd be nice to be able to just confirm it with a gene mutation or a hyperactivation of ApoBec enzyme activity in a genetic test. And actually today, that isn't an easy option, because ApoBec activating mutations is only a small component of what explains it. But this is a slide I added. So for any of you who've got the photocopied slides, this is an additional slide because it just came out in Cancer Research from Li Zhou's group at MGH, in which he inducibly expressed an ApoBec gene, ApoBec3A, which is one of the commonest ones activated in cancer, and it's doxorubin inducible. And what you can see is that when you treat these cells, when it's both doxycycline inducible with an ATR inhibitor to reflect this sensitivity to replication stress, you can see that there's a significant sensitization when the ATR inhibitor is used in the presence of the activated ApoBec. But if you eliminate UNG, which is the uracil glycosylase, that same enzyme that processes these intermediate lesions, you can block that replicative stress. So again, I think there's a strategy for handling these types of DNA repair defects. And this is an area that I think has not received a lot of attention of late, but it is, by sequencing terminology, a very common phenotype in cancer. I want to concentrate the rest of the talk to homologous recombination. This is a pathway of DNA double-strand break repair that acts in cells that are replicating. And the critical step in homologous recombination is taking a double-strand break and processing the ends by digesting the 5' end and exposing a 3' tail, which allows this critical homologous recombination strand exchange reaction to occur, shown here. And this is the so-called D-loop formation. This is where the damaged DNA uses the undamaged duplex, usually the sister chromatid, but it could be any homologous sequence, to allow repair of this broken end by using the undamaged red duplex to repair it. If, however, you get resection, but homologous recombination is defective, you trigger these alternative enjoining pathways that produce deletion alterations in the genome. And this is characteristic of homologous recombination deficiency. So we all know, we heard it in the introduction, that tumors with homologous recombination are sensitive to certain types of DNA damaging agents, the poster child of which are the PARP inhibitors. BRCA1 and 2 mutated tumors almost always have deficiency in homologous recombination. There are multiple mechanisms proposed to lead to homologous recombination deficiency in non-germline associated, which I'll get into. And there are now numerous methods to detect homologous recombination deficiency. So in contrast to what I stated about APOBEC, homologous recombination, we've got numerous ways to pin down the phenotype. So we can exploit the synthetic lethality that I think needs probably no introduction, but it's essentially when you lose the function of the BRCA pathway, the backup pathways can keep the cell alive. But if you then target the backup pathway, the cell will die. But if you have wild type BRCA1 and 2, which applies to the normal cells, targeting that backup pathway has relatively little impact. So that's the concept. So we need to diagnose, and one of the ways to diagnose the defects in homologous recombination was taking these naturally occurring lesion intermediates that are created in proliferating cells when it encounters a barrier, to set up either one-ended double-strand breaks or these daughter strand gaps, which I will come to. But it produces these large-scale structural alterations, which Myriad and many collaborating investigators have found was a strong readout of homologous recombination deficiency. Big changes in the structure of the genome, megabase-sized structural alterations were seen. However, this is data, and I apologize, I've dipped into ovarian cancer briefly, although there are many similarities in this area. But this is from the ARIEL-2 clinical data in which rucaparib was used in relapsed high-grade ovarian carcinoma. And what we're showing here is that the BRCA mutant tumors are doing well in blue. The BRCA wild type are doing not as well. But if you're LOH high, which is this homologous recombination deficiency readout, instead of being where the blue line is, they're still only at the red line, so they're better. But we'd like them to be really a lot better. So can we improve on this? And obviously, my take-home message is the answer to that is yes. And what I'm going to show in the next few slides is that by looking at a combination of these structural variant mutations, which is the basis of the Myriad assay, as well as the single nucleotide mutational signatures from the Sanger group that I introduced at the beginning, combined with homologous recombination gene target sequencing, you can pretty much get to the truth of what is homologous recombination repair deficient, and you can continue to improve this ROC curve for classifying tumors as homologous recombination deficient or not. We've done some additional work which actually looks directly at function in the tumor cells, which I'll describe in a minute. The Sanger group recently in Nature Medicine further improved this ability to diagnose homologous recombination defective cancers by taking whole genome sequencing data and using what they called rearrangement signatures in addition to their mutational signatures to characterize the defect. So if you don't like the details, the take-home message is that the ability to diagnose homologous recombination deficiency, at least based on these genomic alterations, it has improved considerably over the last 5 years. We also looked at functional analysis of homologous recombination by obtaining samples directly from breast cancers. either as biopsies or core biopsies, in which we ex vivo irradiated them and looked for the induction of RAD51 protein foci in the tumor specimens. This is something that can be done, although it was never designed to be a commercial assay. And you can then classify these tumors as repair deficient and proficient, as I'll show in a minute. We can then get this genomic information and then do an integrated analysis. In particular, we looked at tumors that had a high proliferation rate, because sometimes a low proliferation rate could produce a lower level of RAD51, and we didn't want that to be a source of unintended false negativity. And so this is what you can see. Here's as a specimen directly from a patient in a RAD51 proficient tumor. Here are just nuclei dispersed, disaggregated from a specimen. Here is a DNA damaging treatment with 10 gray of ionizing radiation. These are the control cells that show induction of DNA damage with gamma H2AX. And here is the induction of both BRCA1 and RAD51 in a fraction of the cells. It's only seen in a fraction of the cells because it's only in certain phases of the cell cycle will you form these foci. In contrast, a deficient tumor makes DNA damage, but doesn't show this RAD51 or BRCA1 response. And we can categorize tumors as proficient or deficient based on this profile by counting many cells. And the interesting thing from this analysis is that you almost get a bimodal distribution of the cancers that you see. On the x-axis, you're looking at the fold induction of RAD51. On the y-axis is simply the number of tumors. And although this is a small sample, you can see that the deficient tumors and the proficient tumors separate very nicely. And you can see that it is enriched, but not unique to triple negative. It's just over 40% of triple negative, but it's still present in HER2 amplified, as well as even Carlos's ER positive luminal B cancers. So this is a phenotype that's there to be defined in a broad spectrum of breast cancers. And in this recent paper that was published recently, we showed that if you are RAD51 defective, biallelic target gene sequencing defects were prevalent in the vast majority of the deficient tumors, but not the proficient tumors. And you can see this shown here. The blue is deficient and the red is proficient, and there's only one false positive where there's an altered biallelic inactivation of a homologous recombination gene. And similarly, there's only one false negative that appeared to be wild type. And again, if you correlate this with LST, you can't find a perfect cut point that doesn't give you a significant number of either false positives or false negatives. And if you extend this to TCGA data, you can see that although LST population-wise shows a clear difference, it doesn't work at an individual level. We extended this analysis recently published in Nature Communications by reanalyzing the TCGA data sets. And what you can see here is that it confirms this biallelic homologous recombination gene inactivation is a key driver in many, many cancers. Here's ovarian cancer, which is almost the poster child for homologous recombination deficiency, many of which, of course, are germline associated. So the dark is germline, the gray is somatic. And what you can see here is breast cancer, there's a significant somatic component, but in other cancers, and again, staying focused on breast cancer, but this is a phenotype you can see in many cancers. And very interestingly, you can almost categorize pathogenic from variants of uncertain significance by how they perform in this analysis. So we're actually quite interested in the fact that you may be able to, if you see a biallelic hit, but it's a variant of uncertain significance, it won't show the other features. And therefore you can almost categorize them as not significant because it doesn't have the genomic signature. So if you use that two-pronged attack, you can categorize things carefully. These gene defects don't overlap, with a few exceptions. So that helps in the categorization. So now, how well do they predict clinical response? Well, here's the ovarian cancer data Here's the gene mutation carriers, here's the wild type, and this is the survival after platinum. Platinum, really doesn't separate them that well, and probably platinum is not the poster child for homologous recombination, certainly not platinum alone. It's okay, but remember the vast majority of platinum lesions are repaired by nucleotide excision repair. This is prostate cancer, where again the De Bono group at the Royal Marsden in London showed that in metastatic prostate cancer, if you have homologous recombination gene defects, you can start to see a significant benefit from olaparib versus not. So again, this approach may be a best way to handle this targeting problem. There's a bit of devil in the details. This is some recent data from our own group that's just been accepted in JNCI, which is the genetic alterations in ATM-associated breast cancer. Now, this gets categorized with homologous recombination deficiency, but it's not completely clear to me that that's a correct classification, and here's the reason. ATM alterations is almost exclusively ER-positive breast cancer, whereas, of course, BRCA1 associated is very opposite. Even more telling, here are the cases of ATM-associated breast cancer that we identified from this small group. I think there were 24 in total. There's not a single P53 alteration in them. So those seem to be exclusive, which again is very different from what you see in BRCA1 or BRCA2 associated. In BRCA1 associated breast cancer, P53 function is almost entirely lost. BRCA2, a little mixed. So our ability to diagnose homologous recombination deficiency on material available in the clinic today is improving. And the greater use of sequencing, both some degree of genome-wide as well as target gene sequencing, will get you close to diagnosing these deficient cancers. So then what is the strategy? The backup pathways for the BRCA pathway we thought were fairly straightforward, but in fact, there are multiple backup pathways. And it's what's made this field a little more complicated, because there is evidence of backup by both base excision repair, which is certainly where the original idea of how PARP1 or PARP inhibitors were working through short patch base excision repair. But now we've identified at least 3 other backup pathways, all of which may be viable therapeutic strategies for targeting these types of lesions. So one of the questions we've been asking is how can you have 4 different backup pathways? It doesn't make any sense, right? I mean, what's going on? And I think the answer, and again, I'm going to bottom line here, is that homologous recombination deficiency creates a whole bunch of different DNA lesion intermediates. So if you have a 2-ended double-strand break with a sister chromatid in G2, these can certainly be repaired by alternative end joining mechanisms. This may well be the commonest repair pathway. Most of these are probably G2 lesions. However, there's maybe a more lethal lesion, which is where the base excision repair cooperates with lesions on the template strand during replication. They produce these so-called one-ended double-strand breaks. Now, if you have a one-ended double-strand break, the end joining machinery doesn't work because it needs 2 ends. And if you have a one-ended double-strand break, you need a bypass homologous recombination mechanism. And we've argued that RAD52 is that homologous recombination backup mechanism. But it's more complicated than that, because most of these lesions on the template replicating strand get bypassed. Essentially, the replication machinery hops over it and leaves a gap, because then they leave it for the translesion synthesis polymerases to clean up afterwards. So again, that's possibly a mutagenic. The translesion synthesis polymerases are mutagenic, so that may be contributing to the mutagenic profile of these cancers. But it also makes the translesion synthesis polymerases a potential backup pathway target. So my conclusion as to why there are so many synthetic lethal targets are because of these lesion intermediates. The BRCA1/2 pathway is the major pathway that avoids mutations, particularly these structural variant mutations that I've identified. And PARP inhibitors is the best current example of synthetic lethality because they certainly contribute to base excision repair. They may also contribute to the alternative end joining repair pathway. We've looked at other pathways, including this RAD52 dependent pathway that I know we're going to hear a talk on this afternoon. And this was the data we published a few years ago now, in which eliminating RAD52 has a significant impact in a BRCA2 deficient cell, but has no impact in a BRCA2 wild type cell. So it's a classic synthetic lethality, and here is the growth curves showing that there's a complete failure to grow, and it's because almost every cell division is a faulty cell division. And maybe even clearer are these data in which, and we've published almost all of these, if you have a defect in BRCA1, PALB2, BRCA2, the paralogs, they all show synthetic lethality with RAD52. And if you just look at RAD51 foci, that sort of hallmark of this pathway working, you can see that RAD52 on its own does very little to RAD51 function. But in a PALB2 deficient line, it completely eliminates the residual RAD51 activity. So again, demonstrates that this is a valid target, so we've tried to target it. In collaboration with Maria Spies, she developed a crystal structure of the multimerization of RAD52, and certainly looked for drugs that would inhibit this multimerization domain. And in a biochemical assay, came up with a number of drugs that targeted this domain biochemically, and we then validated in control and depleted BRCA1 and BRCA2. And here you can see this is a log scale, no rubbishy IC50s here. This is proper radiobiology. This is a log scale where you get 10 to 20-fold increase in activity. So these are big effects in this context. We took our own approach in collaboration with Bill Holloman and Mike O'Donnell at Cornell and Rockefeller, and screened the Rockefeller compound library and came up again with a series of compounds that target RAD52 DNA binding. And again, this is a log-log scale, so we're looking at significant, at least 10-fold specific sensitization of BRCA2 deficient. This is a CRISPR BRCA2 deficient line in which you can see reproducible dose-dependent sensitization of the BRCA2 deficient cells. And just for comparison, here's mitomycin and olaparib. So again, this puts it well in the order of those types of agents. So although it isn't the greatest structure to target, a library screen of taking that approach certainly seems to at least have revealed compounds at the early stages. We'll see how far we can take these. This is just to emphasize genetically that the translesion synthesis polymerases are a good target. Genetically, if you knock out Rev3, one member of them, in combination with RAD54, you see this very nice synthetic lethality that's specific, whereas the three other combinations grow quite normally. And we've taken this approach with Mike O'Donnell to look at the way that the translesion synthesis polymerases interact with PCNA. And again, taking a target and a biochemical assay that looks at specific translesion synthesis polymerase targeting of that interaction with PCNA. And again, we have some lead compounds from that. Other groups are looking at targeting Pol theta or Pol Q. This was Alan DeAndrea's data published not a couple of years ago, showing that that Pol theta increases homologous recombination in BRCA deficient cancers, and that when you target it, as shown here, with a PARP inhibitor, you get BRCA1 or BRCA2 specific lethality. here shown nicely in this tumor growth curve. So again, a very characteristic picture that you're seeing where you've got specific lethality when you target the donor in the context of a BRCA deficiency. So we've now got multiple examples, including the original PARP inhibitor, again shown as the diagram from the original Nature papers back in 2005, all of which can produce synthetic lethality in these homologous recombination defective cancers. Is there a word of caution? Well, there may be. This is data from Maria Jason's group in Memorial Sloan Kettering, showing that treatment with PARP inhibitors induces genomic instability, whether it's sister chromatid exchanges or total chromatid type aberrations. You can see that every time you treat a cell with olaparib, there's a significant increase. So these are DNA damaging agents. And so that's something, although these are quite well tolerated, they are DNA damaging agents, which means we need to follow them carefully in the future. So to summarize, synthetic lethality in homologous recombination defective cancers might need to go beyond PARP inhibitors. And I think, like everything, we shouldn't rest on our laurels, even though they appear to be very encouraging. I've highlighted the different strategies that might exist, but that we need to be aware of these long-term consequences. So looking for potential other therapeutic options for these tumors is certainly a worthwhile approach. I've highlighted RAD52 because that's my own baby, but I don't mind anybody highlighting any other approach as well. So in overall summary, DNA repair defects are common in breast cancer. Each DNA repair defect has its characteristic genomic landscape, and today that's easy to diagnose, right? $500 whole genomes, it may even come down to $100 if we carry on at this rate. It's really quite remarkable. I've highlighted that in breast cancer, APOBEC and HRD are the two most dominant, other than the aging phenotype. APOBEC, I think, is a challenge for us, and I think I like this approach of looking at ATR inhibitors. Again, they're remarkably non-toxic in human trials. I don't quite understand why they're so non-toxic, but they do seem to be well tolerated. So if we can find the right combination for them, it should be a good approach. With homologous recombination deficiency, I think we've got many options. We know how to diagnose them. We've got a number of options to treat them. And I think this makes a very exciting challenge. We do need more clinical data. And I think the ability to get it now should be there. I think we'll see a lot more clinical data. So with that, I'll just acknowledge lots of people in my lab and collaborators and the various funding agencies who've helped this work happen. It's too many to individually identify, but as we all know, you can't do this on your own. Everything today is a massive team effort. And with that, I'll close. I'm happy to take any questions. Thank you.
Thank you. That was great. Questions?
Jeremy. Oh, it's the DNA atom. It's all my friends coming out.
Great stuff. Obviously, very much a study that we have used to, and the work that you've done has really provided the genetic inspiration for our investigations. It has something to do with your detection technology with RAD51 foci, given that in many circumstances when we were looking carefully at RAD52 foci, focus formation, we found the greatest correlation with focus formation with the inability to repair lesions. So when we saw these incredibly persistent lesions that led ultimately to thousands of fold increases in chromosome loss, that was the condition where we saw 90% focus formation, albeit in yeast. But I'm wondering if that just simply suggests that there is some optimal level of focus formation that's really indicative of repair.
Yeah, you raise a good point. I mean, the way we've used RAD51 is really, if you like, the initial on curve. And we know you need BRCA1 and BRCA2 and probably the other players to get them on. But repair is more complicated than just getting them on. It's getting them on for the right time and then getting them off. And so you can have a repair defective cancer that essentially has persistent damage. And we tend to not study the off curve as much as we do the on curve. They're a bit more, the off curve is more complicated because many things can contribute to it. But the best I can think is that what you're looking at is really these persistent lesions that are, if you like, sucking up these alternative backup pathways, and that's why they persist at 8, 12 hours after your whatever damage you've exposed them to. But I think they accumulate over time. I don't know how reliable it is. We've not studied it systematically. We've certainly got some examples where it's the off part of the curve is the problem. And I think I was just talking to you before the meeting started that we tend to focus more on the front end because there's a lot of action there. But there are defects that occur, if you like, after the D-loop is formed, after the RAD51 foci have been made, and plenty of things can go wrong at that later stage. And so we may need some assays that look at that last part of the reaction.
So as you alluded to some extent with the APOBEC story, when you look at the mutation signatures, it's unclear whether there was a burst of APOBEC activity early in evolution of the tumor evolution or a burst of HR deficiency early in tumor evolution that then is suppressed or adapted to, which won't necessarily inform therapeutic choices. So given that you've kind of combined RAD51 functional assays with mutation signatures, how well do you think the signatures are doing?
Yeah, well, you've raised a point that I've heard many times before, which is the scar that the tumor, the genome reflects what's happened to the tumor at any time in its evolution. And you only need some mutations to get the tumor started, and then they're not needed anymore. And so you could be looking at something that was the past and not the current. And that obviously is one of the arguments why the functional assays are important, because they give you what's happening at the current time in the current cancer that you've got. We're looking into that as to how much, certainly in the RAD51 assay, how much we can see, if you like, RAD51 proficiency when they have the scars, which, if you like, are these reversions. And we know it exists. And so is there a way to diagnose that without having to do these slightly cumbersome functional assays? I think for APOBEC, we don't know quite as much. I don't think we know whether you need an initial burst or not. I don't think I know any lineage data for APOBEC. The biggest problem with APOBEC, which I wanted to highlight, is that there's not a simple genetic, sort of second genetic test to sort of corroborate the signature. So the signature's very clear. Don't think anybody doubts that it's real, but supporting it with an APOBEC assay, APOBEC 3A is the commonest, and 3B are the two commonest ones that we see in human cancer. But you can't account for all of the genomic landscape changes with the target gene analysis. That's what we need to close.
Anyone else? Well, if not, oh, go ahead.
Oh, Gordon.
Polite following your talk. I get to come up next. But just one of the parts that you brought up is around the predictive value of the different tests in the PARP studies that have been done. And these are all ovarian cancer, and I think perhaps to a degree are not informative in that. Most of them, and the ones you showed, are patients who are multiple responders to platinum. That's how they got into the study. And there's a very nice study and work from our group and others that those patients are almost by definition HR defective to a degree. And so now you have trying to predict who is more or less sensitive on a background of already selecting for HRD, which is almost 100% of those patients to a degree. So what do you think is going to happen when we move upfront in breast and ovary? And I think you have a better set of data with the RAD51. What is the real number, and where are we going to get to likely sensitivity here?
Yeah. Well, I take your point about the Ariel studies. I agree with your comments that they are somewhat selected. I mean, the main point I was showing there is that we don't have a even in repeated platinum sensitive, they've evolved. One of the things that's curious, though, is that whenever you look for the prevalence of homologous recombination deficiency, when you go from primary to metastatic, the incidence goes up, doesn't go down. I mean, we've got that data. I didn't show, it's not published. But, you know, the prevalence we saw in the primary breast cancers, which is the published data, is about, you know, 20-ish% of all breast cancer, 40% of triple negatives. If you do that same study in metastatic disease for triple negative, the incidence is 65%. So, you know, the multiple treatment doesn't select out homologous recombination deficiency, which you sort of would expect it to if you're really killing off that cell type, but it doesn't seem to go away. And that's true in the ovarian cancer as well as the breast.
Very quickly. The other, and so I suspect
yeah, the
that there
dead ones.
may be.
I can't test the dead ones, that's true.
Well, no, in ovarian cancer, that's a real problem.
Yeah.
And so the recurrent responders are selecting. That data is now going to be very clear.
Okay, fair point.
So
So you showed ex vivo induction of RAD51 foci with radiation treatment. So a two-part question. Have you shown whether that is a, I assume you've done it in BRCA mutants or not, and can you use this assay, right? Like I show Ki67, right, as a poor man's assay to fishing out the triple negative, the basals that are BRCA competent, but that may share that phenotype.
Yeah, it's not as easy as your Ki67 assay, because if you just look at the basal level of RAD51 foci, it becomes a bit like Ki67. It's a proliferation assay. And so there's much greater discrimination by doing this ex vivo stress, which of course we use radiation because it's an easy way to damage the cells. And it's the stress test that shows whether the BRCA pathway is intact. If you can score, and again, various groups have tried to do this, you can stain even on immunohistochemistry, but it's not the separation, the ability to separate deficient and proficient is not as good because it gets confounded by proliferation.